from Whitesboro High School, and tonight we are going over living environment material. It is only 41 hours until the Living Environment Regents, the last one of its kind, debuts, and it'll be a sad. It's had a good 20-year run. But either way, what we're doing tonight is you guys can call in, you guys can tweet, and we're going to get some questions and go over the things that you are concerned about. First, though, I want to do some thank yous. I want to thank OHM BOCES, especially Scott Morris, who made this happen tonight. I really appreciate it. We'd like to thank Utica City School District, especially Joe Lynch in the booth over there, who is broadcasting for us this evening. We'd like to thank the Whitesboro Science Department for the scheduling of the teachers for tonight. And last but not least, I will not be alone here on the screen tonight. I will be joined by, we have 3 individuals here who are answering the phone. They can't wait to get in front of the camera and showcase their reading skills. We have Madison, Peyton, and Emma, 3 AP Bio standouts who could probably come up here and run the whole thing. They could probably run the whole thing, but instead they're just going to sit at the table and giggle and take your questions. So before we start taking your questions and you guys start calling in, and our number should be going right across this red strip at the top or down here, right there in the corner. You can call in at any time. However, before that, I'd like to take a minute to go over some guarantees, some things that you are guaranteed to see on the Living Environment Regents. I've looked over the past 6 Regences. I didn't want to go any further than that because I didn't want to get into pandemic times. I looked over the last 6 Regences and I broke them down as to what topics come on the most and what you're most likely to see. So I'm going to go through those. So if you are taking this in 2 days, grab yourself a pen, grab yourself a piece of paper, run in the other room, and come scurrying back and start jotting these things down. Because if these things seem unfamiliar, you have questions on them, you absolutely have to call. So I am going to go to this Elmo right now and show you guys what we got over here. And it is popping up right now. There it is. Oh my God. It's like I just conjured it. It's unbelievable. So this is going to be my list of guarantees. So the first thing that we have here is we have food web and energy pyramids. Now, if you take a look, we have a whopping almost 6 questions per test on a food web. Now, this is good news because you've seen food webs in Living Environment and you probably saw them in 5th grade science and maybe a life science in the middle school as well. So this should be pretty easy. But if you are concerned, like, hey, what kind of curveballs might they show us because they're, again, it's not 5th grade material, please call in. We're talking about things like producers and autotrophs, which are huge. We're looking at consumers, and we are also looking at things like what would happen if we removed a species? What would happen to that food web besides it collapsing? If you're not familiar with this shape right here and what an energy pyramid is, you absolutely have to call in. 6 points! It's unbelievable. Can you believe it? Like, it's unbelievable. Why don't they just make a whole test on it? All right, let's keep going. We've got the ecology keeps rolling. Over 4 points, over 4 questions per exam is on human impact on the environment. If you are not familiar with the burning of fossil fuels and or global warming, you need to call in. We can go over that. One of the big things that usually comes up is what is the difference between global warming and the hole in the ozone layer. If you don't know the difference between those two, you cannot rationally think you're going to get 100. You have to know about global warming. Invasive species, red hot. It's been on the exam a ton lately. Very likely you'll see invasive species, generally in a reading section on part C. Next one is direct harvesting. Now this one is like my hot streak one. Like this one's like been like 3 out of the last 4 tests have had a question on direct harvesting. Again, unfamiliar topic, call in, ask what it is. Trade-offs are pretty important. And again, there could be some other things in here like acid rain, things like that. But either way, you're looking right now at about 10% of the exam, if not more, maybe 12% of the exam, is on ecology. Just these 2 topics right here. All right, last one is, I got my shout out there to Mr. Putnam from back in the day who set this up back in the day, experimentation and scientific method. Do you know what a dependent variable is? Do you know what a dependent variable is? What is it? Oh, that's not the definition we're going to use. It changes. That's okay. It's a good thing their mic's off. No, they gave a perfect answer. They said it's what you're measuring in an experiment. Well done. Well done. And it does change. It does change. I just don't like that definition. Again, if we want to clean up these definitions or something like that, you can call in with that. You're kind of right. You're more right than wrong. All right, very good. Sample size is always really big. This one is huge. Scientific explanations are subject to change. Guys, things change. Things change. Like, there's people on this planet who for the longest time thought Michael Jordan was the best basketball player that ever breathed air. We now know in 2024 that's not true. It's LeBron James. Now the Lions are going to start lighting up. Anyone who's watching this is going to call in and complain about that. All right, maybe hypothesis, and then again, a lot of stuff that you'll see on part B1 or part D. So again, those are really big things. You're looking at almost 4 points on experiments. Okay, let's take a look at what other things are going to be on there. We'll go through a couple more. Next one up, natural selection, adaptations. Expect to see the word adaptations. It could be a written question. Natural selection, if you ever have to write about natural selection, the words you want to use are not just survive and reproduce. To win the game of life, it is not about living. It's not about having the most food or the most space. It's all about leaving your chromosomes on this planet. If you've done that, you've won the game of evolution. We cannot forget how important that word reproduce is. Importance of variation and common ancestor. You're looking at about 3 points on natural selection. Let's go through that quick. Immunity. If you are unfamiliar with words like antigen, antibody, antibiotic, these are things that you absolutely want to ask about. These are things that you should know. I would say that vaccines is as close to a guarantee as you can get. A vaccine is very important. You should know what's in a vaccine and how it works so that you don't get sick or you don't get sick a second time. Transplanted organs has been pretty hot lately. We could go over that too. That would be very easy to talk about if we were to talk about antigens and antibodies. Cancer could show up in here, but I find that's more of a mitosis thing than it is anything else. Finally, well, we got a couple more, but real quick, cell organelles. I ranked them by gold, silver, and shameful bronze. We got gold is going to be ribosomes. I think that ribosomes is the most important cell organelle to know. If you don't know what it does, you're probably going to miss a question tomorrow. And it could be written as well, or I'm sorry, on Friday. It could be written as well. Mitochondria, silver, that's pretty good. And then cell membrane, chloroplast, maybe vacuole. I don't know. You could see those. I don't know. Those are hit or miss things. Like now, look, I could sit there and be like, mitochondria, ribosomes, those are hot too. Those are the ones they're going to ask. And then you guys open up your test on Friday, and the first thing, cell membrane. I'm like, well, what do I know? You know what I'm saying? We're just guessing. We're kind of guessing, but we're also using data. You're looking at almost 3 points an exam on cell organelles. Finally, on that list right there, photosynthesis and autotrophs. Photosynthesis, believe it or not, much hotter lately than respiration. Respiration is still a very good chance of being on there, but photosynthesis, the gases that are used and produced, what organelle it occurs in, do you know the basic process? You're looking at about over 2 points in exam on that. I know these things don't sound like much. You're looking at 2 points here, 3 points there, 4 points there. Add these all up, and you're looking at 50% of the exam. I got my last 3. These will be my last 3. Okay. Yuck. Ew. Ew. Protein synthesis. Yeah, just, right? Yeah. Now, the good news is this. Do you have to know fancy words like transcription, translation? Very unlikely. However, you do have to know that DNA codes for RNA, RNA codes for proteins, and if you change that DNA, you are probably going to get a different protein. 2.33 points. And by the way, I have the last 6 Regents exams here. So if somebody was like, hey, there's like one of those protein synthesis things on the part D of, I don't know, August 22, I can look that up for you guys. Last 2, I promise. These will be the last 2. Oh, God. Oh, God. Yikes. Yikes. Oh, put the kids to bed. Put the little ones to bed. We cannot, believe it or not, this is probably the only program where if you call and ask, I'm a little confused as to where babies come from, we actually kind of go over it. Not all the details. You can ask your folks those kind of questions, but we do have to go over the parts. Now, look, females, of course I have here, girls are more important. Everyone knows that. You're like, why are you giving us obvious things? Females bear children. They also have a menstrual cycle. So generally, It's a 9-to-1 ratio. Now, there could very well be a couple questions on male reproductive system on the exam, but you're most likely going to see females. Words like placenta, uterus, if these words scare you, then someone's going to call and ask. Someone's going to call and ask these things, so we have to be mentally prepared for that. Generally, it's on part A and C, and generally placenta is very often being a written question, just so you know. And then to finish out my top 10, and I'll show these again at the end if we have time, you know, at the very end of the program. Diffusion and osmosis, lately, this is all on part D. This is big on part D, and they've been asking a lot about potato cubes, which is interesting because that's an AP biology lab, but they've been doing a lot of stuff on potato cubes and what happens if you soak them in distilled water, what happens if you soak them in salt water, what happens to the mass, and that kind of stuff is asked a lot as well. So to just, in case anybody's dying to see, and you don't have to see, this is my red hot list. There's just some things here that are not like 3 points or 4 points or 5 points, but if we look at them, there are things that have been showing up on 5 straight exams. Gel electrophoresis being one, it's been red hot on part D. Bad news about things like gel electrophoresis and genetic engineering. Those questions could be questions where you have to write, not just about it, but write the actual answer. Like they might ask for what is this genetic technology, and you might have to say gel electrophoresis, which is a mouthful. That's a mouthful. So those are some of my red hot things that are going on, right? Do we have any calls whatsoever or tweets coming in? Not yet. Oh man, well then anything that you, all right, well we'll wait 1 second because guess what, we're probably on a little bit of a delay here, but still, please call in with your questions. We're dying to hear that phone ring. Tweet in your questions. The Twitter or the X, we got to call it X now, right, is down there as well. We're REG_Science Live. So, nothing. Okay, how about this? Do you guys see anything on that list that you think is like, man, these kids, they don't even get, oh wait, we got the phone ringing. Go. Is there anything more after 8? Yeah, you guys take that phone call while I show you. Yeah, we got this. There's what do you call it? Respiration on there. I got biomolecules like DNA and all that kind of stuff. And then I just have some general tips, which I think is important to know. And that is that 8 to 11 questions are just going to be on respiration and, on reading comprehension, data analysis. All right, here it comes. You guys got it? I'm so excited. Who's coming on screen? Yes, here comes Emma Kane. We're going to probably have to kill the Elmo so that we can see you. We'll probably have to kill the Elmo so we can see you right there. Okay, now here's the cool thing about Emma Kane. It looks like she's vanishing, right? It looks like she's vanishing because she's wearing the same color shirt as the green screen behind us. So wait, she looks like, if she was playing Johnny Be Good, she would look like Michael J. Fox in Back to the Future where he's like disappearing, right? All right, we're not going to, you guys, that's a reference that nobody watching this is going to understand, which is a shame. That's a shame. Okay, Emma, what do you got?
We have a question from Mike Stroma from Whitesboro.
Mike Stroma.
They would like you to go over mitosis and meiosis.
Okay, we can do that. Now, Mike Stroma, that's an interesting name because you know what? I'm going to tell you a little something about Mike Stroma. I've never heard that name before, so I'm going to guess it's a kid in the middle school, right?
I bet he's coming up next year.
You think so?
Yeah, you might meet him.
Well, guess what? Maybe I'll see him in AP Bio if my luck is good. Okay, so what was asked was mitosis versus meiosis. Now, I've got excellent news for you. If you had to learn all the stages of mitosis and meiosis, it's very unlikely that you'll have those questions again, like those questions on those specific stages. Instead, the Regents tends to look at more topical things and things that, like what mitosis and meiosis does. So I'm just gonna write down on this thing here, we got mitosis right here, and I got meiosis over here. Now, mitosis. So I always say for mitosis, pretend I'm sitting here and I'm talking, and all of a sudden some guy comes in, and he just busts into the studio. And we're like, we don't even know who this guy is. And he's got a cheese shredder. You're like, what's he doing with the cheese shredder? He starts cheese shredding my face. And there's like pieces of, I know, that's got graphic quick, I'm sorry. There's like pieces of skin going everywhere. And like, of course, everyone's screaming, going crazy. And then we cut to commercial. And then of course, now here's the thing. I'm gonna be, that's a lot for me. That's a lot for me. That's gonna set me back a little bit. But good news. Will I still have open gashes on my face for the rest of my life? The answer is no. That skin will eventually heal. Where do those cells come from? Cell heaven? No, they don't. Why are you don't? They come from mitosis. So mitosis is going to be used for growth, and repair. Now, repair being like my cheese shredded skin. Growth, by the way, everybody who's watching this, at one point you were the size of a grain of sand. Every single person started out the size of a grain of sand. That is a fertilized egg. It was the smallest you ever were. And now look at you. You're amazing, right? So how did you get from that size of a grain of sand to what you are now? Trillions and trillions of cells? Mitosis. Meiosis, is going to make gametes, which is a fancy word for, sex cells. Oh my God, are my kids watching? Put the kids to bed. All right, so it's, put the kids to bed. Sex cells, which are going to be obviously sperm or egg. So just to give you a topical description of mitosis is you're going to start out with a cell with 46 chromosomes. And meiosis, you're going to start out with a cell with 46 chromosomes. So it starts out very similar. Mitosis is going to divide into two cells that are genetically identical to each other. They are going to be identical cells. And we will sometimes say that this is asexual reproduction because it's only one parent. So this is how like bacteria could reproduce. This is how skin cells reproduce. Paramecium could reproduce. It's a type of asexual reproduction. We generally tend to think asexual as being one parent, but also the thing with asexual is it is producing identical cells. Now, if you're watching this and you're like, wait a minute, you forgot to mention interphase and prophase and metaphase and alpha. And look, they could ask the individual stages. The closest thing in the last 6 tests that they have asked for a stage of mitosis is they gave a big picture of a bunch of cells, and some were dividing and some weren't. And they said, draw a cell that is dividing. So if you did not know metaphase or anaphase or prophase, you'd be okay. The key, though, for mitosis is that you would see chromosomes. So anytime you see like any kind of like chromosomes, that would be the case. Now, meiosis is going to divide once, and then it's going to divide again. And what you're going to end up with is you're going to end up with 4 cells. And you're going to have not 46 chromosomes, you're going to have 23, which means those chromosome numbers have been cut in half. And the reason why is because these are going to be sex cells, so I could turn them into, if you're like, oh, he made balloons. Nope. Guess again, they're sperm cells, right? And those could be used to combine with the egg cell, and the 23 and the 23 recombine. So what could they ask for meiosis? The key for meiosis, if I can slide this up just a little bit, would be that we are getting different cells. Thank goodness, right? We're getting different cells. Because think about it. If all of you, and I know nobody wants to think about this, but look at, man, this is just the way things are. If all your parents' gametes were the same, mom's eggs, dad's sperm, you and your siblings would be exactly the same. And the beautiful thing is that you are wildly different from your sibling, unless, of course, you have an identical twin. And the reason for that is because even though it's like dealing cards, you've got 46 cards here, you're going to get 23. But if I deal 23 to one person and 23 to another, it's chances are you're not going to get the same exact cards. So you're going to have different things. One thing I will add in to this that I think is very important if you're watching is lately on the exam, they've been huge on the word recombination. And they're using that word for fertilization, meaning when a sperm and an egg combine, they're calling it recombination. So keep in mind that mitosis, you are looking at things like growth and repair. You're getting genetically identical cells. It's an asexual type of reproduction. And meiosis, you're going to be getting 4 cells, but you're getting a lot of cells with differences. You're getting different chromosomes. Now, if you were to call back and be like, he didn't go over metaphase 2, you don't have to know all those details. You're looking at the big picture process. All right, I feel like we had some action over there. Do we have another call? Yeah. All right, here comes Madison. Madison is, go ahead. Madison is not disappearing because she wore a white sweatshirt as opposed to the green sweatshirt. But the green sweatshirt's fun, right? I almost wore a green shirt today. That would be weird if like the entire time you would not be able to focus because you're like, my God, he's like, I'm just seeing right through him.
Okay, Rhonda from Camden wants to know what...
Rhonda from Camden, okay, go ahead.
She wants to know what body systems does she need to know for the test?
Okay, so it sounds like we have a question, and it is a question from the Blue Devils. All right, wait, is Camden the Blue Devils? Yeah. Congratulations to the Camden girls softball team winning their Section 3 title. That was pretty good, right? You know, we got a Section 3 softball title in the room as well. You know, I'm not trying to brag, but I'm just saying that, you know, good job, Camden. Wait, I forgot the question. What was it? Oh, the body systems? Yeah. Sorry, I got caught up thinking about Central New York softball. All right, anyways, so here's what we're gonna do, body systems. Okay, the biggest one you have to know is circulatory. So the good news is this, like, okay, so my son's taking this test, and he said to me, he's like, hey, I'm like, what'd you do in class today? I'm like, what'd you do in class today? He's like, we got to ask any question that we're worried about for the test in 2 days. I'm like, that's awesome. I'm like, what'd you ask? And he asked like this very specific question about a nephron, which is like something that's found in the kidney. And I was like, oh, I'd stop you right there. I mean, it's great that you know this information and all these body parts. However, again, the Regents is very topical. Like, if you can name every single intricate part of a body system, but do you know what it does and what its main purpose? The reason why I have circulatory down on the list is because, which I think is really important, I'm just gonna grab a different colored marker right here, is because circulatory is really big because it comes into the, it deals with the making connections lab, where you guys had to like squeeze a clothespin, you had to take your pulse rate, things like that. Not sure if you remembered that lab. You're supposed to do it, but either way, circulatory, the big theme of circulatory is that it's gonna move materials. And no offense, the one that they are always obsessed with is oxygen. This usually shows up on part D. They'll ask why your heart rate increases during exercise, and you cannot just say the blood moves faster. That is not an acceptable answer. You have to say that we're moving materials like oxygen and sugar at a faster rate. So circulatory would definitely be one. Respiratory. Guys, you took physio.
We did.
What do you think the respiratory system does? Breathing. So when I'm breathing, what's that going to put into the blood? Yeah, it's gonna diffuse oxygen, and what's it gonna remove?
CO2.
Carbon dioxide. Very good. That's why they're the best of the best over here. It's gonna diffuse CO2. You're probably not mic'd up right now, but their answers are killer, man. They're killing it. Right? Very good. The other two that you're maybe gonna see, I don't think you're gonna see, but you could. It could very well be asked. Is the digestive system and excretory. Now, again, my son was asking me a really specific question on the excretory system, and I was like, man, nephron, not going to be. In fact, I'm saying the word nephron. Some people at home are like, what? Like, they're like, I mean, you know, kudos to any teacher that goes over that kind of stuff. I love it. But excretory is going to be big for removing wastes. Thanks, Maddie Manley. And digestive system is obviously going to break down nutrients, and those are really the big four that you should know, and I put them in order of importance. I put them in order of importance. Circulatory, moving materials, very likely to see it. Very likely to see it on part D, about moving oxygen, moving sugar, or moving wastes away from cells after they build up. Respiratory system is going to put oxygen into the blood, and then digestive excretory maybe. You could see endocrine or nervous, but those are going to be cell communication. And if someone were to call in with a question on endocrine or the nervous system, I would probably cut right to a cell receptor question. All right, did you guys are all talking over there? You got one? Yeah. Okay, that one was a quick one because there's no sense in me going over the entire circulatory system when they're not going to ask like what the vena cava is and stuff like that. Do you have more than one question?
No.
Oh, that's okay. I like when we can pick. I was hoping we'd have like 7. All right, Madison Manley.
Miles wants to know what's the difference between DNA and RNA.
I'm sorry. Did you go to school?
I forgot it.
Oh,
It's a middle
Miles.
schooler. I'm sorry. I'm sorry,
Well,
Miles.
it doesn't matter. We're good. We're good. No, it's okay. Just whatever. Just don't do your job. Whatever. No. All right. Anyways, no, it's fine. Miles. Hey, at least you got the first name. You know.
I was going to put Miles for Miles Middle School, but then
That's not going to work. So the question was DNA and RNA.
Yes.
Okay, so this is huge. This is big. So what you're going to need to do for DNA and RNA is you're going to have to go over. You're going to have to know what DNA is, what RNA is, and what's the whole purpose of it. So this is going to deal with something that we call protein synthesis. And proteins are nutrients that are made up of. Proteins are made up of amino acids. Amino acids, very good. They're made up of amino acids, and that amino acid sequence is going to be determined by your DNA. So what you're going to have is you're going to have DNA that is going to be in the nucleus, tucked away in the nucleus, 6 feet of DNA per cell. You got 6 feet of DNA, that's as tall as I am, 6 feet of DNA per cell, it's crazy, right? And that DNA is not going to be changed into, but used to produce RNA, which can go out into the cytosol or the let's make this easier, let's say the cytoplasm, right? So the DNA is going to be not turned into, but used to make RNA, and that RNA is going to determine what we call an amino acid sequence. All right, and that's going to be like the order of amino acids that you guys have. Now, of course, if you look over at somebody and they've got blonde hair and somebody that's got brown hair, the reason why their hair is 2 different colors. Now, if I said, why is your hair 2 different colors? Most people are going to say, well, it's because we have different chromosomes or different DNA. And that's correct, but really it's the different DNA that's ultimately coding for a different amino acid sequence. So some people have thinner hair, thicker hair, straight hair, curly and wavy, and all these are because you have different proteins. Now, of course, your DNA codes for your proteins. So the question that we had is, what's the difference between DNA and RNA? They both use bases, right? They both use bases. I'm sure we're used to the DNA shape that looks like this, right? RNA is more linear, like this, but the big difference is not double-stranded versus single-stranded, even though that's a pretty big difference, is the bases. So for DNA, G, C, A, T, T, A, C, G, C. Those are all DNA bases, right? RNA bases are not the same 4 letters. Which letter do we not see in RNA? We don't see the T. Instead, we've got the letter U. So the way we always match these up is we say, gangster cat always tripping. We say gangster cat always tripping because gangster cat, G and C, always tripping A and T. And then when I teach this in class, I have a picture of gangster cat. It's awesome. It's a real cat. He's dressed up. It's pretty fun. But anyways, gangster cat, G's and C's go together. So if I had to match up this 3-letter triplet of GCA, gangster, G matches up with C, cat matches up with gangster, and here's the problem that we have with RNA, there is no T. So A matches up with U, right? We say for RNA, gangster cat's always understanding, even though my students are always like, gangster cat's always urinating. All right, T can match up with A, T matches up with A, and A matches up with U. Cat, gangster, gangster, cat, cat, gangster. Those letters match up in that way. Again, the big difference being, of course, is that there are U's. Now, of course, if we change one of these letters, let's say we change this T to a C, that's a mutation. Uh-oh, got a mutation here. That A is now going to match up, and that A is not going to be there. The C will now match up with a G, because G's and C's go together. And now what's going to happen is this 3-letter sequence is probably going to code for a different amino acid. So instead of having, amino acid A and we'll say amino acid C, this one now would be like amino acid. Maybe it was supposed to be B and it's now like D or something like that. So it's going to be a different amino acid. So anyways, the main difference between DNA and RNA, I would say make sure you can match those up. If we have time at the end, I'm not sure you guys got another call, right? Which we'll get to. You got a couple questions, great. I was going to say towards the end, if I have some time, I would love to show. I got a bunch of different regions here. I bet I could pick up one at completely random, maybe not this one, and show you guys a question like this about how to do that kind of stuff. But either way, the main difference: make sure you know the basis and be ready tomorrow to go from DNA to RNA. And here's another thing: make sure you can spot DNA. How do we know for DNA? Gangster cat. Always tripping, RNA, gangster cat. He's always understanding, right? There you go. All right, you guys ready? Who's coming out this time? Oh, brand new person. Our third person. If you're wondering, like, how many people are here? Peyton Leahy, AP Bio monster, knows everything. She could teach it. Ready? I'm taking a break. Go ahead. Go ahead. You got it. You got it. You got it. No, I'm just kidding. I'm right here. All right, so ready? What's the question we got?
This is a question from Kaylee Davis from Whitesboro High School.
Kaylee Davis! I know Kaylee Davis. Kaylee, good luck on Friday. Go ahead.
And she wants to know if we can go over osmosis.
Yeah. Yes, it's going to be on there. It's good. Wasn't it one of my guarantees was diffusion osmosis? Okay, let's start with the basics. Let's start with the basics that you guys should know. This is tough for me to say on live TV. You know what I'm saying? But I'm going to open up here. Open up. When I was a little boy, I was a little chubby boy. This is true. I was a little chubby boy, and I had a mullet before they were fashionable, and glasses. Yeah, it wasn't good. And I used to live on the top of this really big hill, Westmoreland Road, right? Which I'm not sure if anyone in Whitesboro is familiar with. And I used to get on my bike right here, and I would ride down the hill to a Nice and Easy that is now a pizzeria. I can't say the name because we're not sponsored by anybody. You know what I'm saying? Yeah, I don't want to put all. I don't want these other pizzerias to get mad at us. I like, whatever. But either way, it's a pizzeria now. It used to be a Nice and Easy, which is a gas station. And I would ride down the hill, and I would get all sorts of delicious treats to eat because I loved candy, because I was a portly little boy that liked candy. So here's the thing. All right, when I went from this position here, high to low, when I went down the hill from a high altitude to a low altitude, did I have to pedal the bike? No, oh my God, are you kidding me? I would have never been able to stop. If you got that mass moving, I would have never been able to, I probably would have burst into flames like a comet entering the atmosphere. No, it would have been impossible. So what happens is there's going to be no energy used. And I use this in class every year to talk about diffusion because, or they sometimes call it passive transport, but you're more likely going to see diffusion. And this is going to be diffusion because diffusion is moving from a high concentration to a low concentration without any energy. And the question that Kaylee had was not specifically on diffusion, even though it's always nice to know high to low, but hers was on osmosis. And osmosis is a very specific diffusion of water. It is the movement of water. So what has the Regents been asking about lately where we can talk about osmosis? The big things they've been talking about, oh God, oh man. Did I spell potato right, or is there an E at the end? You think I'm good? Nice. Okay. So potato, they've been talking about cutting potatoes into cubes. So meaning like you have these cubes like this, right, of these potatoes. And they say if you soak this cube in distilled water, which we're getting calls, man, this is going to be crazy. All right. Distilled water is 100% water. The question they've been asking is what's going to happen to this cube? Well, we know this. Water is going to go high to low. This is the high concentration. And if you're like, how do I know that's the high concentration? Because it's 100%, man. There's nothing that you can have higher than 100%. Sometimes you'll hear people be like, man, this kid always gives 110%. That makes no sense. It's 100% is the most. So if we know that that is 100% water with nothing else in it, we don't know what the water content is inside of here, but we know it's not 100% water because it's potato. There's potato parts. So where's the water going to go? The water is going to diffuse into that cell, and what's going to happen? It is going to cause the cell or the potato cube to gain mass because water is going to come rushing in. Keep in mind that potatoes are plants and they have cell walls, and those cell walls will let water come in, but it won't cause the cell to explode. So the cell will actually start to puff out, right? And what happens is all that water rushing in causes that potato to gain mass. So what's the kind of stuff that you see on the Regents exam? They'll give you a question like this, right? Now, look at this. They got beaker 1, 2, and 4, and they're talking about, again, I could have looked right there for the word potato. It was right there on the sheet the whole time. But I'm glad I got you guys to help my spelling. But what happens here is when you put the potato in distilled water, just like the picture I have, it gained 4 grams. That means water is moving into the potato. That is osmosis, right? Now, you guys did something similar in lab with like a little bag. You made like a little baggie, and you probably put like starch in it and sugar in it and soaked it in water. But this would be a potato cube. Now, you'll notice, let me switch colors here. I'm going to switch to a nice purple. We've got 16% salt solution. Well, salt is going to have a very low water concentration. So what is going to happen here? If we put a potato like this in salt water, and we know that that is a low concentration of water because it's mainly salt, now there's a higher percentage here, the water diffuses out, and it lost mass. So generally the questions that they've been asking have looked like this. I'm not saying they're going to put the same one on there on Friday, but it would be lovely if they did, because then you could be like, hey, I watched that weird guy on TV, and he told me all about the potato cubes. He couldn't spell it, but he did his best. And you could see that right there, that water would rush either in or out depending on the concentration. So that is the big thing with osmosis. The key, though, is don't forget your roots. Don't forget that diffusion of water is osmosis, and it's moving high to low. Again, you do not have to pedal downhill. You can just coast downhill, and you'll be good. Guys, how are we doing on questions? You got a couple? All right. All right, very good. Oh, it's Madison Manley again. Here she comes. You just appeared out of nowhere.
This is a similar topic. Abby Calhoun from New York Mills wants to know.
Mills!
Yes.
Go Marauders! Go Marauders.
She wants to know if you can go over diffusion through a membrane.
Yes. That's all she asked was diffusion through a membrane?
Yeah.
Ooh, I wish we knew if it was the lab or she wanted to talk.
Yes, the lab. It was the lab.
Let's do it. Let's do it. Okay, very good. That's a perfect question to ask. Okay, so, well, it was Abby Calhoun from New York Mills? Okay, very good. Well, Abby, they have not put a question like this on in a while. They've mainly been going over the potato cubes, but you know what? Maybe the potato cubes have had their run and they're going to try something different. I would love to go over diffusion through a membrane. There's some takeaways here that we could see. So what you did in diffusion through a membrane is you guys had to make this little bag. And in the bag, you put two things: sugar or glucose and starch. Okay. Now, if you don't recall, starch is a long chain of sugars, right? It's a huge chain of sugars, and it's really, really, really big. Now, the whole point of this is that sugar is small, it's tiny, and starch is big. So, I always compare this with a chain link fence. You and your friends are sitting on opposite side of a chain link fence, and I give you guys each a bucket of golf balls, and I say, whip these at your friend as hard as you can. Right now, of course, don't do this. Right? Could you throw a golf ball, and could it go through a chain link fence, and could it hit your friend right in the face? The answer is yes. Why? The golf ball's small, and those little chain links are big enough for a golf ball to go through. Then I say, let's get rid of the golf balls and replace it with bocce balls, which if you haven't seen bocce balls, it's like lawn bowling. I said, okay, now you can't toss it over the fence. You got to whip this at your friend. Can you hit the friend? Now the answer is no. Why? The bocce ball is too big to go through the chain link fence. So that is the theme of this lab. What you guys did in this lab is you tested the water on the outside. Sugar should have gone through that cell membrane. Why? It is small. I know you can probably not read my writing, but trust me when I'm saying that says small. The starch cannot go through. It is too big. So what we should find is sugar should be out here like this in the water, but we don't see any starch coming out. So how do we know starch stayed in? Well, one of the things that we put inside the water is something called iodine. Now, you guys, I got news for you. They never ask iodine on the Regents exam. Instead, they call it starch indicator solution. I have no idea why they just can't say iodine. It's not that big of a deal. But they say starch indicator solution. Great. Well, anyways, what happens is iodine or starch indicator solution is nice and small. So what does that do? That goes through the bag, and the inside of the bag turns purple. So the inside of the bag turns purple, and the outside does not. So what does that mean? Iodine went in, starch stayed put. It stayed right where it was. And that is the big theme. What do they usually ask about on the Regents? In the last 6 Regents that I looked at, the biggest thing they've asked is the iodine or the starch indicator solution. They like to ask, how do you know a positive reaction took place, or how do you know there was a presence of starch? And you would have to say the starch turned purple or it turned bluish black. Different teachers say different things. But either way, again, the inside of the bag turns purple because iodine went in. Starch is too big. It's like trying to throw a basketball through a chain link fence. It's just not going through, right? Guys, I think I tried to do my best. Back-to-back diffusion questions. Yeah. Part D, we should have, between those two things and the DNA, RNA thing, Part D is looking like it's getting covered, right? Okay, here comes Section 3 softball champion. She doesn't even want to talk about it. She's like, I don't even want to talk about it. Me, little old me. Section 3 softball champion, Emma Kane.
Sarah from New Hartford would like to know about electrophoresis.
Oh, man. Sarah from New Hartford. Go Spartans. You know, as a Whitesboro person, I probably shouldn't be saying go Spartans, but my brother and sister both teach at New Hartford. Mr. Ryan and Mrs. Roth. They're excellent teachers, right? Math and English. But either way, I'm glad we got a New Hartford question, right? That I've already forgotten. What was the question again?
Okay.
Electrophoresis, that's a good one, too. Holy cats. Holy cats. Okay, so watch this. Wait, you guys have a question? Watch this. Wait, I'm gonna do a social experiment right now. Maddie Manley, come up here for a second. Ready? Ready? You can kill the Alamo for 1 second. There you go. Ready? We're gonna go over gel electrophoresis, but I want to just prove a point. Ready? Maddie Manley.
Yes.
I want you right now on TV. I'm gonna say some characteristics. I want you to picture an animal.
Okay.
It has fur.
Okay.
It has a long tail.
Okay.
It's got pointy ears.
Okay.
It's got 4 legs.
Okay.
What are you picturing?
German Shepherd.
Ooh, sorry, the answer was a fox. Beat it. All right, so now wait. So there's a point behind. Wait, no, you can really. No, there's a point for. Wait, there's a point. There's a reason I did that. The reason why is because when it comes to describing an organism or finding out like if organisms are related or whatever, you can go with physical characteristics or you can go with a different type of thing, and that is molecular evidence. So molecular evidence, and this has been a big theme on the exam lately, knowing that DNA or proteins is the best evidence to have that 2 things are related. So how can you test if 2 organisms have similar DNA? Right? Because again, there's a lot of animals that have fur, and I'm not doing that to make a fool of you. There's a lot of animals have fur and a long tail and stuff like that. So let's go to gel electrophoresis. So what they do for gel electrophoresis, and this is what the pictures usually look like, they give you like a rectangle that looks like this. And at the top of that rectangle, they give you like these little spots like this. Of course, it's drawn way better than what I have. In fact, I just use something like this that I already have right here. There, that looks way nicer than my horrible drawing I did. Okay, and let's just say that right here, we have a crime scene. Oh no, somebody committed a crime. And I've got 3 suspects, Emma, Peyton, and Maddie. One of them committed a crime. So what we do is we take their DNA. So what we're getting here each time is the DNA from those individuals. Write down DNA every single time. And that DNA is placed inside of a gel. Now, before that DNA is placed inside of a gel, it is cut. What do we cut DNA with? Scissors, no, too big. Needles, too big. Lasers, too big. They're going to be cut with, do you guys remember this as enzymes? They're going to use enzymes to cut that DNA. And because everybody's different, unless you have an identical twin, because everybody's different, your DNA is going to cut up differently. So what they then do is they run the DNA through the gel. And everyone's DNA is going to be cut differently, and it's going to separate into these different fragments. So what you're going to have is you're going to see these different fragments here separating out. So if we look at the crime scene, which is right here, and I said to you, which person, E, P, or M, committed the crime, you would just match up the bands. Maddie Manley's matching up the bands right now, and it is, well, it's not Peyton, but that one's pretty close, right? That one looks pretty close. But either way, that's what they would do. Nobody committed the crime in this case, right? There's nobody, not any of these 3 suspects at least, right? But they match up. Now, here's something that's a little bit different about gel electrophoresis that you should know. Again, what you're doing here, the big thing is you're comparing DNA sequences. But what they've been saying lately on the exam is they're not saying DNA sequences, they're saying bases or base pairs. They're using a specific thing like the A's, T's, C's, and G's. But wait, there's more. Another thing they've been doing lately in the last 6 exams that they might not have done 10 years ago. So if you're working out of an older Regents book or an older Regents review book or something like that, what you might see that's different is which of these bands, which of these chunks are the bigger chunks. And generally what happens is the chunks at the top are usually your larger ones, and your ones at the bottom are your smaller ones. Why? Because smaller chunks of DNA can move easily through the gel. Keep in mind, these are like slices of Jell-O. And there's little tiny openings or pores in these gels. And what happens is the DNA can kind of push through, and if it's smaller, it pushes through easier. I always say it's like picture having a guy you're on level ground, and you got like a 300-pound guy in a shopping cart. He's like he's like laying in the shopping cart. And I said, without running, just using your arms, push that thing as far as you can. 300-pound guy, you might give it all your might and push it, and he might only go a few feet. Now replace the 300-pound guy with a Chihuahua, like a little tiny dog that's barely like anything, right? And you put that in the shopping cart, and you push that. Oh man, that Chihuahua's going flying, right? Why? Because it's smaller and it's easier to push. It's the same theme here. So you might have to know not only which things match up with which, but you might have to know the chunks, which ones are smaller and which ones are bigger. Keep in mind the smaller ones are usually at the bottom, they go further. Something too that they would have asked years ago and they haven't asked lately is what charge are we moving towards? And we're always heading in a positive direction. New year, new me. We're leaving the negative behind. I'm moving towards the positive. I don't know if you'll see that, but you will absolutely see gel electrophoresis. Now there's one more thing I want to do before I end gel electrophoresis. We got another question coming up soon? Excellent. Well, we got a couple. Good. All right, it's okay if we go over too, right? This is one of those things where when you answer the question, they might ask you to write it out. This is a nightmare situation. They might, on part D, give you a picture like this and say, what's this process above? And if you're like, man, I cannot remember, write down gel and then try getting a word as close to electricity as you can. But this is one of those things where they will sometimes ask you, what is this process? So it might be something you want to commit to memory. I know my writing's a little bit gross there, but that is gel electrophoresis. And I guarantee if I picked up any Regents, look at this. I'm just picking up the last Regents, it's just January 2024. Boom, take a look right there. Big question on gel electrophoresis. Again, they ask it. Just go on the internet, type in living environment Regents, click on any exam, look at part D of the last 6 exams. You'll probably find a gel electrophoresis question. Great question. Was that the New Hartford question? That was the New Hartford question, correct? I forgot the name. Sarah, nice job. Sarah, that's a good one to go over. Great job. Good job, Spartans. All right, you guys got another one? Yeah. Who's coming up? All right. We got Peyton Leahy walking up here. Peyton Leahy. What do we got going on? Sorry, I'm just trying to organize all my crazy papers here. Yeah, I'm good.
I have a question from Brittany from Whitesboro.
Brittany from Whitesboro.
And she wants to know, what do ribosomes do?
Oh, this is easy. This will be a 10. You guys got another question? Yeah. Good, because this will be 10 seconds. This will be 10 seconds. Okay. Yeah, we'll keep this one super short and sweet, right? In fact, you know what? I'm going to take the gel electrophoresis picture I had, yoink, turn it right around. That's all the space I need. That's all the space that I'm going to go over. So ribosomes, it is very simple. It's very simple. However, I will add the little tweaks that you should probably know. Ribosome, that is going to make what? Proteins. Exactly, very good. However, what might they say instead of proteins? They might say enzymes, because enzymes are made out of large proteins. They could say hormones, but they probably won't. They could say antibodies. They probably won't. Well, they could. But enzymes, or they might say chains of amino acids. Now, you do have to know that proteins are made out of amino acids. So the very simple thing about ribosomes is just know that they are used for synthesizing proteins. Ribosomes very likely will be on there. And just for kicks, the other one that they ask all the time is mitochondria. If you were to have a written question and to write down powerhouse of the cell, your teacher will crawl into the fetal position and start crying on the floor because they probably told you 1,000 times, don't ever say powerhouse of the cell. That's like for internet memes and things like that. Instead, what you're going to say is Powerhouse of the cell. Just kidding. Respiration. You'll say respiration, or you can say it's producing ATP. So those, I know that nobody asked about, Brittany didn't ask about mitochondria. Yeah, but what the heck? We might as well throw it on there. It only took 30 seconds. Those are the two big organelles that you should see on the I got AP. Well, yeah, that one too, but the living environment regions. You guys got another one? You look like you guys look like you're losing steam. Are you guys okay?
Oh, yeah, we're great.
Come on up.
No, I got it.
Look at Maddie doesn't even need the paper anymore. At first she thought she was gonna need papers every single time.
Okay, I have a question from the Matt Ryan
Oh God, Matt Ryan.
from Whitesboro. He wants to
Good
know,
for him. I'm glad he's watching. He's being forced to. So.
He wants to know how immune cells, specifically white blood cells, attack foreign antigens.
Ooh. Well, I'm gonna say this: the fact that he said how white blood cells attack foreign antigens gives me great satisfaction, because the key is, do you know the difference between an antibody and an antigen? Now, the big thing for this is an antigen is going to be a protein found on a cell surface, or really just, an antigen is really just anything that really might not belong. I mean, our cells have antigens that we recognize, and we're very good at recognizing our own, but we're also very good at attacking antigens that we don't use. So for example, when I teach this class, I always think antigens like a jersey. And I'll sometimes have my students close their eyes and I'll put on like a hockey jersey. When I open their eyes, I'll be like, okay, what am I wearing? Like a hockey jersey. I'm like, great, what team? And then, we kind of go through that, like, how do you know? Because you recognize what it looks like, right? So he wanted to know how white blood cells attack foreign antigens. So let's just say it's a foreign antigen, which means it is something that we want to kill, like a viral particle or a bacteria cell, or one of the big things on the Regents lately has been a transplanted organ, like getting somebody's kidney, and those antigens of that kidney don't match the other person, which is why if you are going to get an organ from someone, it is probably going to be a relative. Why? Because the proteins they have are similar to the proteins you have because you're related to them, right? Anyways, so white blood cells are going to do two different things for foreign antigens. The first thing is it could eat them or engulf them, which is a process called phagocytosis, which I do not think, hey, you high-fived each other. You guys knew the term? Good for you guys. The great news is I don't think that term would be on the Regents, but they should certainly show a picture. They could certainly show a picture of something engulfing something, right? The other thing is the white blood cell might produce antibodies. So if you learned about things like T cells and B cells, those are great to know. But those are probably pretty detailed for this exam. You'd be more likely to know that you are going to make antibodies, and those antibodies are going to attack antigens, the things that we don't recognize, like the jerseys that we don't recognize, right? Now I got to add something in here, right? As much as I like to go over the different white blood cells and what they do, the eating and engulfing, very unlikely to see. Antibodies, very likely to see. But what is the most likely thing you are going to see? And that is going to be what a vaccine is. A vaccine should absolutely be on this test, and it'll probably be a written question. If you get the flu shot, what are they giving you? They're giving you the flu. They're monsters. No, I'm just kidding. They're giving you a weak or dead version of the virus. And when you get it, it is weak, you are strong, so you beat it up real easy. And what do you produce? You make antibodies, right? You make antibodies, which will, like we said up here, attack the foreign antigens. So every year I get the flu shot, right? I get the flu shot, and every year they give me a dead version of the flu, and hopefully it's the strains that will be prevalent in December, January, February. My body makes antibodies. Now, of course, the thing is I'm going to go get a vaccine every single year because, as we know, the flu, the common cold, they mutate very often. And again, the medical community just predicts which strains will be most likely out there. But again, you're training your body by making antibodies, right? That's a big thing. So make sure you know what a vaccine is. It is an injection of a dead or weak disease. Your body attacks it. You make antibodies. You should not have to know things like, for physio, do you have to know like lymphocytes? No? B cells, T cells, any of that stuff? Okay, good. Well, guess what? You don't have to know that stuff for the exam. If your teachers are teaching it, though, kudos to them, man. I love when students are over-taught these things because it could be very easy to just kind of give the basics, but that is the big takeaway. And one of the things I do want to add here is proteins. And again, these are like proteins are going to be foreign invaders that we are going to attack. These things are going to be things that we want to get rid of. All right. You guys got another one? Man, we're flying through. It's 7:58. I can go a little bit over 8. I don't care. It doesn't bother me, right? As long as people have questions, as long as we have like legit questions coming up. Emma Kane, you're disappearing. Yep. Yeah.
Maddie from Clinton would like to know a little about global warming.
Clinton. I forget. Clinton. Good for them. I can tell you something about Clinton. Their U12 softball team's pretty solid. They put a hurting on us a couple weeks ago. Right? Whitesboro Gray did not fare too well. That's okay. We're playing them again. We're hoping it goes well. All right. What was the question?
Global warming.
Oh, man, that's a really important thing to go over. Holy cats. All right, so global warming is something you absolutely have to know. Sorry, I started thinking about the softball stuff. All right. So global warming. I'm going to start with this. If you do not know what these two words are, when I say these two words, I'm writing down 3, but I'm really focusing on these 2. If you don't know those 2 words, you're going to get a couple of questions wrong on this test, man. You're not going to get it all correct because you do have to know burning fossil fuels. So when you burn fossil fuels, and if you don't know what a fossil fuel is, you could be watching this and be like, not exactly quite sure. We're looking at things like gasoline, like how I got here, oil, and coal. Those are big fossil fuels. Like natural gas might be another one too. You might not necessarily be like gasoline, but whatever. Either way, these are fossil fuels. When you burn fossil fuels, I can barely speak. When you burn fossil fuels, what you're going to produce is carbon dioxide. And that is something that you have to know. And that should not be crazy. Because right now I'm standing here and I'm burning sugar as I'm sitting here talking to this camera. I'm burning sugar. And one of the things I'm breathing out into this room is carbon dioxide. Generally, whenever you burn anything organic, CO2 is probably going to be produced. So to make CO2 is not crazy. What that is going to do is that is going to blanket the earth. And when it blankets the earth, it is going to trap heat. It is going to trap heat inside the earth, and that's going to cause the average temperature to rise. So what are we looking at here for the big connection? Burning fossil fuels, the average annual temperature is increasing. And they are going to take this into a variety of different ways. What they are going to do, I'm going to call it right now, is on part C, there will be a little passage that you read. I'm just saying it because they do this all the time. This is not anything shocking. On part C, they'll give you a little passage to read, and they'll talk about I don't know, how it's getting warmer up in Canada, and that's affecting the rabbits because the rabbits usually blend in with the snow, and there's less snow coverage. This is one that I'm going over that was recent, right? Hotter temperatures, not that much snow. Even if you think about it, like even this past winter, I know you shouldn't look, when you're talking about global warming, you should never look at one location for one season. That's absolutely goofy. But we didn't get a winter that was crazy this year. Didn't get a ton of snow. So you would think that like white rabbits that normally blend in with the snow or the rabbits that turn white in the winter would be at a disadvantage. They'll give you something like that. Or they'll talk about the warmer temperatures in the ocean are causing these fish schools to travel and disrupt food webs. They'll go over that stuff all the time. So just be prepared to know this. The other thing I'm going to throw in there, I know nobody asked it. I don't care. I'm going over it. Is I'm going to draw a dark red line right here. And I'm separating fossil fuels and global warming. This is global warming right here. It's getting hot. The other side, hole in the ozone layer. All right, I'm going to ask you guys questions. I know you're taking phone calls. I don't care. I'm going to interrupt you. Right? Hole in the ozone layer. What's the ozone layer block us from?
Sun rays. Yeah.
Sun rays? What sun rays?
That'll be on
UV rays. Oh, God, I made a U and a V, and they look exactly the same. You got UV rays. Very good. Do you know what the UV rays, if they hit us, what it could cause? skin cancer, and
Burn.
sunburns, yeah. But it can also cause mutations too. Yeah, who knew? You probably didn't see that one coming. And so this is the question. Nobody ever knows this. If the three of you, we got 3 people here who could probably teach this class, right? They crushed AP Bio. They made a mockery of it, right? They're operating at a completely different level. But watch this. Watch this. I'm not doing this to embarrass you. Do you guys know what chemicals cause holes in the ozone layer? CO2. CO2 is not correct. CO2 is over here for global warming. See, I'm telling you, this is something. Look at, you got 3 students over here destroy everything bio-wise, and everyone struggles with this. Greenhouse gases, that is also over here for global warming. Yes, this is typical. I'm not using you as an example to ridicule you on television. I'm going to ask you to stop talking. I'm going to have it'd be great if you guys could just shut your yappers. It's CFCs. CFCs. No, I'm telling you this. You got 3 people over here that had probably your average in AP Bio collectively was probably like a 99.7. So, and again, this is something that affects everybody. Everybody knows a lot of people are good with CO2 causing the temperature to rise. But what causes the hole in the ozone layer? It's CFCs, right? The good news is this. You want the good news? Very unlikely that you'll see ozone compared to burning fossil fuels. Now, if you're like, oh, good, ozone layer won't be on there. Oh, no, it could be. But I know this. This is a could be. Hole in the ozone layer is a could be. This is a definite. This is a definite. You're going to see that for sure. Now watch me be wrong. Watch the first question tomorrow be on hole in the ozone layer. And I'll be like, oh. All right. You guys got another question? We got 8:04. I'll probably go to like 8:15. You know. You got 3 questions. Ooh, you know what? I might be at the point now where I just kind of want to like hear what you have and go over them. But you know what? Let's hear what the first one is. Maybe the first one's going to be too good.
Madison from Nyack
Wait,
Middle
everyone that's
School.
calling's name is Madison. Is this?
The other girl was Maddie.
Oh,
This is
Maddie.
Madison.
Well, okay, that's way different. All right, what do you got?
She's from Nyack Middle School, and she would like
No
you-
way. Is that for real?
Yes.
Oh, that's awesome. Wow, because I mean it's being streamed, so Nyack, it's
Yep.
a little far away from our viewing area. What do you got?
She would like you to go over some biochem.
Okay, that's awesome. I'll do that. It's a very great. I love the general questions where I can kind of have a little bit of leeway here. All right, so that's excellent. So I will go over them in order of importance, and I'll go over them pretty quick. So in terms of, she wants biochem. Okay, well, I'm going to tell you this. The biochem won't be too crazy, but there are some themes that we're going to see. Now, normally, I'm going to do something different here. I'm gonna write down this. I should write down proteins first. Lately, I know, gasp, who knew? Right, he's not writing down proteins first. DNA is probably asked about more lately. They've been asking a lot that DNA is made up of individual bases, and those bases are gangster cat, always tripping, like we said, if it's DNA, or gangster cat, always understanding if it's RNA, G, C, A, and U, if it is RNA. Lately, there have been a lot of questions on which bases match, what DNA is made of. They have been focusing on DNA lately. I almost put it on my hot list, but I didn't. The next one that I think is super important to know is protein. So again, this is like what we call a polymer over here. This is polymer, and this would be like a monomer. And monomer is a fancy word, so I should probably write down a much easier word, which is subunit. They like to say subunit instead. So proteins are made out of amino acids. That's really, like, so if I was doing this, the last time I did this show was 5 years ago. If this was 5 years ago, there is no way I would have started with DNA. I would have started with protein first. DNA has been asked about a lot lately, so I feel pretty confident going with that one first. Either way, this is like 1A and 1B in terms of importance. They're both really important. Next one that I would say I should know would be starches, which are made up of glucose, or you could have said simple sugars, whatever you want to say. But that would be the monomers or the subunits of that. Starches and sugars are important because it comes up in that lab that we talked about, the diffusion to a membrane lab that we had probably about a half an hour ago. And then I don't think it's going to get too crazy after that. I think that what you should know is these 3 things. The only other thing I can think of for biomolecules that you could see is going to be, and I'm going to put a little line here because it actually is one of the one above, and that is enzymes. You should absolutely know that enzymes have a specific shape. I always draw enzymes to look like Pac-Man, right? I do it all the time, right? That enzyme has what something is called an active site. And that active site, you might not have to know that term, but you do have to know that there is a very specific shape on that enzyme. And if you change that shape, that enzyme is not going to work. Now, why did I draw the line between the enzymes and the other three above? Enzymes are large proteins. So really, they're kind of the same. They're made up of long chains of amino acids. So here's a question for you. Ready, guys? You want to try this one? Or you guys, do you guys not want to answer any other questions since the greenhouse, or not they don't know who you are. That's okay. Not from NIAC, they're not going to know who you are, right? Okay. So we're going to talk about how this protein, this enzyme, could be denatured, which means it changes shape. Do you remember the two? High temperature. Thank you for not saying temperature. High temperature. Very good. And?
Something with the pH. I don't know if it's higher.
Both. Any change. You're good. See, I told you guys know everything, right? If you have a high temperature or high or low pH, so acidic or basic, that could cause the shape of the protein or the enzyme to change. Oh, that does not look at all like a Pac-Man, right? This is really, you're useless, right? You're not going to be able to do your job. And why? It's because, again, that active site has a specific shape. So this is my way of teaching why it's high temperature and not low temperature, right? Ready? This will be common knowledge, right? You go to the store and you buy a steak. What color is the steak? Red. Pink or red? That is a great answer. You take that pink or red steak and you throw it on the grill. What color does that steak turn? Brown or gray? That is a great answer. What happened to the protein of the steak? It denatured. Okay, I go to the store, I buy that red steak. I put it in the freezer. Not a high temperature, a low temperature. I take it out of the freezer a day later. What color is it? Same color, red or pink, right? Low temperatures will not change the protein structure. Slapping that steak on a grill, oh, I'm not even on the frame. Slapping that steak on a grill and using heat, you'll see those proteins change. It's the same thing with eggs. You buy eggs, the egg white is clear, you throw it in a frying pan, it turns white. Keep it in your fridge forever, it still stays clear, right? So proteins, it's really high temperatures. Denaturation or changing of the protein shape, very likely to be on there. I'm glad that we had that call from NIAC because this part up here seems like I really kind of babied people by being like, okay, here's your biochemistry, but this enzyme stuff is super important. And remember, another word for enzymes, a great word for enzymes, are catalysts, which means they are going to speed up reactions. Yeah, Peyton's coming up. Peyton Leahy. Peyton Leahy's coming up. She's got the microphone. Oh. Okay.
I have a question from Sean from Whitesboro.
That's me. Somebody's using my name. It's a fake name.
Yep.
Do you know who it really is?
It's you. It was you.
Okay, I'm calling from the future? Okay, That's excellent. Yes.
And he wants to know about gene expression.
Ooh, anything specific on gene expression?
That was it.
Just gene expression. Okay, I'll turn this into two different things real quick. All right, so what should you know for gene expression? All right, we'll do a couple things with gene expression. So, okay, ready? So one of the things that you're definitely going to see is something about genes in the environment, meaning what they're going to say is you're going to have, here, I'll draw a bunny. There, isn't that cute? It's pretty good, right? So you got a rabbit, and in the warmer temperatures, the fur of that rabbit is going to be brown, right? And in colder temperatures, that fur of the rabbit is going to turn, you can probably guess. Yeah, you got it. It's going to be white. It's very similar to what I said just earlier on global warming, right? So here's the thing. Did the rabbit's DNA change? Did it mutate? No, its DNA is the same, right? The DNA is the exact same as it started, right? I don't know how many chromosomes a rabbit has. I don't know. I could probably say anything. People believe it, 30, I don't know, whatever. They got 30 chromosomes, right? Those 30 chromosomes, which I'm totally making up the number, would be the same whether they're in the warm or the cold, right? So what do we see here? We see that it's not the genes that change, It's the gene expression, right? And we see this too. There's people that are watching this who, when they are exposed to UV light, their skin gets darker and their hair gets lighter. And are you going to sit there and be like, oh, I got a glorious tan? Oh, my DNA changed. Your DNA didn't change, right? You're producing more melanin, or you're producing more pigment because of the exposure to UV light. But again, the big thing is the DNA is the same. This is one thing about gene expression you should know. The other thing that they like to ask about gene expression, I'll draw a line, is they'll give you like a cell. They always give the same 3 cells. Like, here's a nerve cell, right? So you got a nerve cell or a neuron, right? You guys remember how many chromosomes we have in a regular body cell? 40. You got it. Nice job. See, I feel like you guys have picked up a ton of steam since the other questions. Then they'll give you like a muscle cell. Here's some muscle fibers. You guys probably know all about this from physio. You probably know about like sarcomeres and stuff like that. You know, and actin and myosin proteins and all this kind of stuff. All right, either way, muscle cells, 46 chromosomes, and then they'll give you like a blob, like here's a skin cell. It's pretty lame. It's like, looks like a cloud. Like, this is a skin cell, 46 chromosomes. So you're looking at muscle, nerve, and you're looking at skin. And one of the things that they will ask, let me see if I can move this up a little bit. One of the things they'll ask is, well, they all have the same 46 chromosomes, right? They all have the same 46 chromosomes. How come they look so different and act so different? One's a nerve moving impulses, one's a muscle that's contracting, one's a skin cell. It's just like, just sitting here, I'm a skin cell. Those 3 things have the same DNA. Why are they so different? They use different parts of the DNA. You do not need to use all 6 feet of your DNA per cell to build a nerve cell. You might need parts of one chromosome, parts of another. So different cells use different genes. So even though we have the same 46, we have to know that different cells use different genes, right? And that is something that could come up as well. That is that's on my my guarantee list as well. I think that stuff tends to go on there. It tends to be a multiple choice question, and it's usually on part B1 or 2 or something like that. All right, guys, what do we got left? We got one final question. This will be good because it's 8:15. I feel like I'm like, plowing through here. All right, so Madison Manley's going to end us for the night, probably, right? Probably, right? We look like we're closing down shop. Yep. You guys are just picking up the phone and hanging it up. Like, we're closed. All right, what do you got to end the night?
Carly from Whitesboro wants to know.
Oh man, you think this is Carly Hughes?
I think it is Carly Hughes.
Oh man, if this is Carly Hughes, you're the best. Right?
She wants to know if you can go over energy flow in the food web.
Oh man, that's a great one to end on. All right, so I know neither of you guys had me for living environment, but wait, she wants food web and energy flow? I love this question. Okay, so here's the thing. I'm actually going to show you something. I'm not making the guarantee this year. But in the past, hold on, I'm coming back. In the past, I brought the picture. In the past, I would make outrageous claims that if you do not see this shape on the living environment Regents, then I would shave it in the back of my head. And one year, I think it was 2013, I made this claim for years, for over a decade. I was like, if you don't see the energy pyramid on the living environment exam, I'll shave it in the back of my head. And then one year, it wasn't on there, and I had to shave. I know you can't see. I had like 90% more hair, by the way, at this point. But I shaved it. Oh, you can't see anything, can you?
No, you can't see it.
Can you see it?
A little bit.
Can I trace it a little bit? People thought it was candy corn. Oh, that doesn't help at all. How about this? Nope, does not make things better. Yeah, maybe green? Said the girl with the, no. You think so? All right, well, anyways, people thought it was candy corn. Like, why do you have candy corn shaved in the back of your head? Oh, my wife was super livid. She was not pleased at all. Now I'm so zoomed in on this, I'm gonna zoom back out a little bit. There you go. So this is something that you might have learned in like 5th grade, and I'm not going to say like, this is why are you asking this question, you're going over in 5th grade, because there is a way that they tweak this to make this more difficult. So first of all, this is going to be like some kind of plant, right? But they're not going to say plant. They might use a picture and give you a picture of a tree, but they're probably going to use a word like producer, or they might give you a word like autotroph. Autotroph being something that you should absolutely know. One of the things we did not go over, and we should have, is photosynthesis, but whatever, it's kind of getting late. But producer or autotroph. And I always say, like, this is always the biggest, thickest part of the energy pyramid, and I can prove it. I always call this the throughway test. Let's say you're in the car, you're driving on you're not driving, you're just riding in the passenger seat of the throughway, and your phone dies, so you have to look out the window or do something else. So your phone's dead. You're like, oh no, this is torture. And you're looking out the window. What living things do you see when you're driving on the New York State Thruway? Grass, trees, shrubs, trees, grass, trees, weeds, grass, trees, right? Hawk. Grass, trees, shrubs, grass, trees, forest, deer, right? Like every now and then you'll see like cow, right? Or like that weird woodchuck thing. What's he doing? He's just standing. Like you'll see those things sometimes, but most of the time, like even right now, if you go and look out your window, you're going to just see mostly green. So the biggest level is always your producers, which they ask, you know, they could say producer, they could say autotroph. They love to show a food web and say which one of these is an autotroph, and you have to make sure you pick a plant. The next level up, let's go with a bunny, because I'm so good at drawing them. Now, will they say bunny? No, they'll probably say something like a consumer or heterotroph. Or they could say primary consumer, herbivore. There's a lot of words. Yeah, did you say herbivore first? Yeah, they can use a lot of different words. I mean, you don't know what you're going to get, and this is where they make it they make it somewhat difficult, where they give you guys these things. Last one, what's going to eat a bunny? Sure. Oh, a fox is fine. We've got a fox in there. So now a fox can be called a consumer because it's eaten to anything that eats is a consumer. Now we're looking at the difference of a primary and secondary consumer. We could also call it a heterotroph. But more specifically, we could call it a carnivore, right? ¿Dónde está mis niños de español? What is the word for meat in español? Carne, right, there you go. So there you go. So that they can give you a bunch of different words for this. Here's where things get a little bit more difficult. One of the things we see is that it tapers as we go up, so we're going to get less available energy as we go up. Why? It's because energy is going to be lost as heat. That little bunny that goes hopping around the forest is going to burn sugar and release heat to the environment. So that heat, in fact, only 10% can transfer to the next thing. So like a McDonald's cheeseburger. Oh, we shouldn't be naming brand names. I told you we weren't going to name brand names. Just a cheeseburger from a random place that has gold arches, whatever. That has like 290 calories. The most you can keep from that cheeseburger is probably like 29 calories. You can only keep about 1/10 of it, right? The 90% of the energy is going to be spent running around, doing the things that you're doing, even just sitting there zoning out watching this. Your brain's using a considerable amount of glucose doing that. So that is what we're going to have for the energy pyramid. They like to ask, the questions that they might have on the food webs are really pretty easy. You should be okay on those. However, read the questions carefully. Read the questions carefully. Sometimes they ask very different kinds of questions. Make sure you're looking at the diagrams correctly. They love to ask if you take out the wolves, what's going to happen to the rabbits. Make sure you're following the arrows. Whenever they give you guys a thing like this, if they have like a tree, and then they have a caterpillar, right? And then they have a bird like this, and they give you that thing. Keep in mind that arrow means it's going into the organism's stomach. So the tree is going into the caterpillar's belly, and the caterpillar is going into the bird's belly, right? That's generally the flow of energy that we would have, okay? All right, it is 8:21. Real quick, I'm just going to throw up here on the screen my guarantees. Please, I'm going to back out a little bit on this so that you guys can see. Please make sure that you know these things for tomorrow. Nobody called on experiments. That's okay. That makes me feel all right. Dependent variable, independent variable. Would have loved to go over dependent variable. It's really what you're measuring in the experiment. Y-axis type things, it's usually on the y-axis. A lot of people, no one called in on independent, dependent variables. We did, however, take care of burning fossil fuels. Ooh, direct harvesting. That's when you're like over hunting, over logging. You're taking too many things out. Keep your eyes peeled tomorrow for direct harvesting. What do we do on the next page? No one asked natural selection. That's okay. The key word is adaptations. Follow adaptations. Remember, survive and reproduce. The secret of life, leave your chromosomes on the planet. Immunity, we tackled a little bit. We especially got vaccines. We got antigens versus antibodies. We got our gold and silver cell organelles. People called in on that. We didn't do anything on photosynthesis, but we got autotroph, and we did get the energy pyramid in there, so I feel pretty good about that. And then finally, maybe it's because the girls screened, oh, protein synthesis, we got that. Reproduction, we didn't get any questions. No? Yeah, maybe that's for the best. You know what I'm saying? Maybe I'm not like gonna lose sleep over that tonight. And osmosis, we absolutely crushed it. Those are the top 10 things you're most likely to see. You guys want to come back out and say goodbye? You know, you can bring the microphone if you want. Can we, there's no way to change the background, right? Like you can't change the background, right? Well, you are disappearing. There you go. Let's see. Well, let's see if they can change the background. That'd be great. Thank you, thank you, by the way, to these three for answering the phones all night. Thank you to Scott Morris over at OHM BOCES. Thank you to Joe Lynch, who's been working over there. He's like going from the Elmo. He's cutting the Elmo out and all that kind of stuff. And it's a shame that Emma Kane's disappearing. She is vanishing because she's wearing a shirt that matches the back. All right, that's okay. All right, guys. Well, we are all set. Thank you guys so much. Good luck on the Regents. Yeah, and if anyone tweets any questions, I'll try to answer them tonight if I can. All right? All right.